Welcome back friends. Welcome back again to one more of the lecture session from this channel. So in this lecture session we will be studying regarding role of biotechnology in medicine from biotechnology and its applications in detail. So here there are various recombinant DNA technological procedures which have made an immense impact in the area of healthcare by regulating the production of many number of effective therapeutic drugs. So here these effective therapeutic drugs, they are capable to function so efficiently without inducing any immunological responses. So here the specific immunological responses which are most commonly observed in case of the specific therapeutic drugs which are obtained from the non-human sources, such specific immunological responses they are not observed in case of the recombinant therapeutics. And at present nearly 30 recombinant therapeutics they have designed worldwide, and out of which 12 of the recombinant therapeutic drugs they are being marketed in India. Is it clear? So let me write it on the board. There are various recombinant DNA technological procedures which have made an immense impact in the area of healthcare by regulating the production of effective therapeutic drugs. And here these effective therapeutic drugs, they won't induce any immunological responses. So here the specific therapeutic drugs which are produced from the non-human sources, they may induce certain immunological responses. But here the specific therapeutic drugs which are produced from the recombinant DNA technology, such specific recombinant therapeutic drugs, they won't induce any immunological responses. And at present nearly 30 recombinant therapeutic drugs they have designed worldwide, and out of which 12 of the recombinant therapeutic drugs they are being marketed in India. Is it clear? At present nearly 30 recombinant therapeutic drugs have been designed worldwide, and out of which 12 of the recombinant therapeutic drugs are being marketed in India. And then after we will be studying regarding genetically engineered insulin. So here what is genetically engineered insulin? Is it clear? So let me arrange the things here. And then after we will be studying regarding genetically engineered insulin. So here what is genetically engineered insulin? Maintenance of adult onset of diabetes mellitus, it is possible by intaking the human insulin at regular intervals. So here most commonly within the human body, insulin secretion takes place at regular intervals. And for suppose if the insulin hormone secretion, if it does not take place within the human body, under such condition, the sugar levels within the blood of the human body, it may increase to the elevated levels. And here, it may lead to the causes of a specific metabolic disorder termed as diabetes mellitus. So here, in order to overcome the problem, in general, insulin hormone is injected externally on the surface of the abdomen. So here the specific insulin hormone which is injected externally on the surface of the abdomen, it is obtained or it is extracted from the non-human sources like slaughtered cattle and pigs. So here the specific insulin hormone which is most commonly obtained or which is most commonly extracted from the non-human sources like slaughtered cattle and pigs, they are capable to function so efficiently likewise the normal human insulin without inducing any allergic reactions. Is it clear? The specific hormone or the specific insulin hormone which is obtained or which is extracted from the non-human sources like slaughtered cattle and pigs, they are capable to function so efficiently without inducing any allergic reactions. In most of the cases, the specific insulin hormone which is obtained or which is extracted from the slaughtered cattle and pigs, they are capable to function so efficiently without inducing any allergic reactions. Only in case of very rare people, the specific insulin hormone which is obtained from the non-human sources like slaughtered cattle and pigs, they are capable to induce certain allergic reactions. Is it clear? So let me write it on the board. Maintenance of adult onset of diabetes is possible by intaking the human insulin at regular intervals. When insulin hormone secretion does not take place within the human body, it may lead to the causes of a specific metabolic disorder termed as diabetes mellitus. Is it clear? When insulin hormone secretion does not take place within the human body, it may lead to the causes of a specific metabolic disorder termed as diabetes mellitus. So here, in order to overcome the diabetes mellitus, most commonly the insulin hormone, it is obtained or it is extracted from the non-human sources like slaughtered cattle and pigs. And here these specific insulin hormone which is most commonly obtained or which is most commonly extracted from the non-human sources like slaughtered cattle and pigs, they are capable to function so efficiently without inducing any allergic reactions. Is it clear? Insulin hormone which is most commonly isolated from the non-human sources like slaughtered cattle and pigs. cattle and pigs are capable to function so efficiently without inducing any allergic reactions. Without inducing any allergic reactions. So here you make it a small clarification. Why the specific insulin hormone? Most commonly it is injected externally on the surface of the abdomen. Instead of intaking the specific insulin hormone orally into the body, why the specific insulin hormone? Most commonly it is injected externally on the surface of the abdomen. So here the answer to this question is very simple. As we all know, insulin is a peptide hormone. That means it is composed of amino acid residues. So here, when the specific insulin hormone, instead of injecting externally on the surface of the abdomen, if we intake the specific insulin hormone orally into our body, the specific insulin hormone, it may undergo degradation by the means of specific degradative enzymes like proteases within the digestive system of the body. So here, once when the insulin hormone, once when it enters into the digestive system of the body, within the digestive system of the body, there are many number of degradative enzymes like proteases. So here these proteases, they are capable to degrade the specific insulin hormone. So the reason why most commonly the insulin hormone, it is injected externally on the surface of the abdomen instead of intaking orally into our body. Is it clear? Insulin hormone. Insulin hormone most commonly it is injected externally on the surface of the abdomen. On the surface of the abdomen instead of intaking orally into our body. Instead of intaking orally into our body, because insulin is a peptide hormone. Because insulin is a peptide hormone. Peptide hormone which undergoes degradation. In the presence of degradative enzymes like proteases. Degradative enzymes like proteases. Is it clear? So let me arrange the things here. And if you look at the structure of the insulin hormone, so here as already mentioned, insulin is a peptide hormone. That means it is composed of 51 amino acid residues. And here the specific insulin hormone or the recombinant insulin hormone, it can be successfully produced within the bacterial cell itself. And here the specific insulin hormone, likewise the other enzymes and other hormones, it also synthesizes within the human body in inactive form. And here the inactive form of the insulin, it has to undergo processing event in order to obtain the mature form of the insulin. Is it clear? So let me repeat here. Insulin is a peptide hormone. Peptide hormone which is composed of 51 amino acid residues. 51 amino acid residues. And likewise, the other enzymes and hormones, insulin production within the human body, takes place in inactive form. And here the inactive form of the insulin, it has to undergo processing event in order to obtain the mature form of the insulin. Is it clear? The specific inactive form of insulin has to undergo processing event in order to obtain the mature form of the insulin hormone. Right? So here let me show you how the processing event takes place within the human body in order to obtain the mature form of the insulin. So let me show it here. So let us assume it as the inactive form of insulin. And here the inactive form of insulin, it has to undergo processing event, right? Processing event. So here once when the inactive form of the insulin, once when it undergoes processing event, here we can obtain the mature form of the insulin. So here within the mature form of the insulin, here there is an availability of only A peptide sequence and B peptide sequence. But here there is no availability of free C stretch of the peptide sequence. Is it clear? During the processing event, here there is an availability of only the mature form of the insulin. So here the mature form of the insulin, it only contains A peptide sequence and B peptide sequence. So here the A peptide sequence and B peptide sequence, they may remain in association by taking the advantage of disulfide bond. And here during the processing event, successful cleavage of free C stretch of the peptide sequence takes place. Is it clear? So let me show it here. So here it is the A peptide sequence and it is the B peptide sequence. Is it clear? So here both the A and B peptide sequence, they may remain in association by taking the advantage of disulfide bond. So here it is nothing but what? Disulfide bond. And here successful cleavage of free C stretch of peptide sequence takes place. Is it clear? So let me write it here. During the processing event, both the A and B, A peptide sequence and B peptide sequence, peptide sequence may remain in association with disulfide bond, and here during the processing event the free C stretch of the peptide sequence, it undergoes successful cleavage here. Is it clear? After the formation of the mature form of insulin, successful cleavage of free C stretch of the peptide sequence takes place. C stretch of the peptide sequence takes place. Here, one of the most important challenge of various recombinant DNA technological procedures here it is nothing but to assemble the mature form of the insulin. So in 1983, Eli Lilly, an American company, they have designed two gene sequences which are very corresponding to the human insulin. So here after designing the two gene sequences which are very corresponding to the human insulin, then after they have inserted the specific gene sequences into the vector DNA of the E. coli host. Now here, likewise the other genes of the vector DNA, even the foreign desirable gene, it had undergone or it undergoes successful expression within the E. coli host. So here after the successful expression of the two gene sequences within the E. coli host, then after they have obtained the mature form of the insulin, and here they have joined the two peptide sequences by taking the advantage of disulfide bond. Is it clear? In 1983, Eli Lilly, an American company, have designed two gene sequences which are very corresponding to the human insulin. After designing, so let me write it here. After designing the two gene sequences, they have inserted the two gene sequences within the E. coli host. The mature form of insulin have obtained by joining the two peptide sequences by disulfide bond. Is it clear? So here this is regarding genetically engineered insulin. and then after we will be studying regarding gene therapy in detail. Is it clear? So let me arrange the things here. And then after we will be studying regarding gene therapy. So here what is gene therapy? So here the most important objective of gene therapy here is nothing but to treat a disease. And here basically gene therapy, it involves many number of strategies. So here what are the various strategies which are a part of gene therapy here? Gene therapy basically it involves the identification of the defective genes within the cells and tissues of an individual. So here after identifying the defective gene within the cells and tissues of an individual, now here the defective gene, it has to be replaced with the correct gene. So here once when the defective gene, once when it is replaced with the functional gene or with the accurate gene, now here the functional gene, it is capable to compensate the non-functional gene. Is it clear? So here all these are the various strategies which are a part of gene therapy. So let me write it here. The most important objective of gene therapy is to treat a disease. So gene therapy basically it involves many number of strategies, and these may include identification of defective gene within the cells and tissues of an individual. So here after identifying the defective gene within the cells and tissues of an individual, now here the defective gene, it has to be replaced with the functional gene. Is it clear? Defective gene has to be replaced with the functional gene. In the cells and tissues of an individual. So here all these are the various strategies which are a part of gene therapy which successfully occur within the particular living organism. So here gene therapy, it can be successfully organized under two different strategies, and these may include enzyme replacement therapy and the other one is bone marrow replacement therapy. So at first we will be studying regarding enzyme replacement therapy. So here enzyme replacement therapy, for the very first time it was carried out in the year 1990 to a 4-year baby girl who was suffering from a specific immunological disorder termed as severe combined immunodeficiency syndrome. Is it clear? Gene therapy can be carried out under two different strategies. And these may include enzyme replacement therapy, and the other one is bone marrow replacement therapy. Bone marrow replacement therapy. Replacement therapy. So at first we will be studying regarding enzyme replacement therapy. So here enzyme replacement therapy, for the very first time it was carried out to a 4-year baby girl who was suffering from a specific immunological disorder termed as severe combined immunodeficiency syndrome. Enzyme replacement therapy. Enzyme replacement therapy, for the very first time, it was carried out in the year 1990 to a 4-year baby girl suffering from immunological disorder, severe combined immunodeficiency syndrome. So let me write it here. Severe combined immunodeficiency syndrome, which is simply abbreviated as SCID. So here most commonly within our body, when the adenosine deaminase gene, so let me write it here, adenosine deaminase. So here when the adenosine deaminase gene, once when it works out so efficiently, under such condition, the B lymphocytes and T lymphocytes, they are capable to function so accurately within the body. But here, when the adenosine deaminase gene, once when it remains in inactive form, under such condition, here B lymphocytes and T lymphocytes, they won't work so efficiently. So here, once when the B lymphocytes and T lymphocytes, once when they won't work so efficiently, it may lead to the process of a specific immunological disorder termed as severe combined immunodeficiency syndrome. Is it clear? Adenosine deaminase gene, when it remains in inactive form, B lymphocytes and T lymphocytes do not work so efficiently, and here it may lead to the causes of severe combined immunodeficiency syndrome. So here, in order to overcome the problem, here we need to inject the adenosine deaminase gene within the cells and tissues of an individual. So here, once when we inject the adenosine deaminase gene within the cells and tissues of an individual, so here the successful expression of adenosine deaminase gene, it takes place within the cells and tissues of an individual. So here, once when the adenosine deaminase gene, once when it undergoes successful expression, here B lymphocytes and T lymphocytes production takes place, so that it can eradicate the occurrence of severe combined immunodeficiency syndrome. Is it clear? So let me write it here. Adenosine deaminase when it remains in functional form, B lymphocytes and T lymphocytes production takes place, and T lymphocytes production takes place. But here, when adenosine deaminase gene when it remains in inactive form, SCID occurs. Is it clear? So here we need to replace the inactive form of the adenosine deaminase gene with the functional gene. So here once when we replace the inactive form of the adenosine deaminase gene with the functional gene, here the specific functional gene, it is capable to work so efficiently in order to eradicate the severe combined immunodeficiency syndrome. Is it clear? So let me arrange the things here. So here during gene therapy, successful replacement of the functional gene takes place. Functional gene takes place in order to eradicate SCID, severe combined immunodeficiency syndrome. And the other alternative strategy through which gene therapy occurs, here it is nothing but bone marrow replacement therapy. So here what exactly happens within the bone marrow replacement therapy? Here it is nothing but initially the specific lymphocytes from the blood of the patient, they are successfully isolated. So here after isolating the specific lymphocytes from the blood of the patient, now here the specific lymphocytes, they are allowed to grow within the culture medium. So here after growing up the specific lymphocytes within the culture medium, now here a specific retroviral vector in association with the foreign desirable gene, it is allowed to insert into the lymphocytes. So here after inserting the foreign desirable gene into the lymphocytes by taking the advantage of retroviral vector, now here again the specific lymphocytes, they are retransfused into the blood of the patient. And here the specific strategy, here it is nothing but bone marrow replacement therapy. Is it clear? In bone marrow replacement therapy, successful isolation of lymphocytes from the blood of the patient is isolated. So here after isolating the lymphocytes from the blood of the patient, these lymphocytes, they are allowed to grow within the culture medium. Is it clear? These lymphocytes are allowed to grow within the culture medium. Now a specific retroviral vector in association with foreign desirable gene, is allowed to insert into the lymphocytes. Okay. these specific lymphocytes, again they are retransfused into the blood of the patient. And here this specific strategy, here it is nothing but bone marrow replacement therapy. And here the problem with both of these specific strategies, here it is nothing but they are not completely curative. Instead of taking the advantage of these two specific strategies at the adult stage, if we take these specific strategies at the early embryonic stages, it could be a permanent cure. Is it clear? The basic problem with both of these strategies is nothing but they are not completely curative. They are not completely curative. But if we inject the specific desirable gene during the early embryonic stages, it could be a permanent cure. It could be a permanent cure. Is it clear? And then after we will be studying regarding molecular diagnosis. So here what is molecular diagnosis? So let me arrange the things here. And then after we will be studying regarding molecular diagnosis. So here what is molecular diagnosis? In order to regulate or in order to provide the effective treatment of a disease, two things are mandatory, and these may include early diagnosis and the other one is studying the pathophysiology. So here early diagnosis and studying the pathophysiology, it is very important in order to provide the effective treatment to a particular disease. So here by taking the advantage of various conventional analysis like urine analysis and serum analysis, here we can suspect or here we can predict the presence of the disease causing pathogen only at the severe stage but not at the initial stages. Is it clear? By taking the advantage of various conventional analysis like urine analysis and serum analysis, here we cannot predict or here we cannot suspect the presence of the disease causing pathogen like bacteria and virus at the initial stages. Here we can predict the presence of the disease causing pathogen like bacteria and virus only at the severe stages. So here we cannot predict the presence of the disease causing pathogen like bacteria and virus at the initial stages by taking the advantage of various conventional analysis like urine analysis and serum analysis. So here in order to overcome the problem, there are many number of versatile techniques through which we can detect the presence of the disease causing pathogen even at the early stages. And these may include recombinant DNA technology, polymerase chain reaction, and the other one is antigen antibody interaction. Is it clear? So let me write it on the board. In order to provide the effective treatment of a disease, early diagnosis and studying the pathophysiology is very important. So here by taking the advantage of conventional analysis conventional analysis like urine analysis and serum analysis. Urine and serum analysis. We cannot predict the presence of disease causing pathogen at early stages. So here, in order to overcome the problem, there are many number of versatile techniques. So here, what are the various versatile techniques here? These may include recombinant DNA technology, polymerase chain reaction, and the other one is antigen antibody interactions. Is it clear? There are many number of versatile techniques through which we can detect the presence of disease causing pathogen at early stages, and these may include polymerase chain reaction, and the other one is antigen antibody interactions. So at first, if we look at the polymerase chain reaction, so here most commonly polymerase chain reaction, it is used for amplifying the disease causing gene. So here, by taking the advantage of polymerase chain reaction, here we can detect the presence of HIV virus, and even the specific polymerase chain reaction, it is also used in order to detect the cancer virus within the individual's body. Is it clear? So let me write it here. Polymerase chain reaction is most commonly used to detect the HIV virus in AIDS patients, and even it is also used to detect the presence of cancer virus in cancerous patients. Is it clear? So here, now let us have a look how the specific polymerase chain reaction, it is most commonly used in order to identify, or here in order to amplify the disease causing gene. So initially, a single stranded DNA or single stranded RNA with a radioactive probe, it is successfully placed within the individual's body. Is it clear? So let me show it here. So here it is nothing but what? Single stranded DNA or single stranded RNA which is in association with radioactive probe. Right? So here it is nothing but what? Radioactive probe. And here let us we assume it as the disease causing gene. So let me write it here. Disease causing gene. So here, if there is disease causing gene within the individual's body under such condition, here the single stranded DNA or single stranded RNA which is in association with the radioactive probe, it does not undergo complementarity or it does not undergo base pairing, and even here radioactivity nature it is not observed. Is it clear? If there is disease causing gene within the individual's body under such condition, the single stranded DNA or single stranded RNA which is in association with radioactive probe, it does not undergo base pairing with the disease causing gene, and even the radioactivity nature it is not observed. So here absence of base pairing and absence of radioactivity nature, it indicates that disease causing gene it is present within the individual's body, and here appearance of radioactivity nature and appearance of base pairing in between the single stranded DNA or RNA in association with the concerned DNA, it indicates that here there is no disease causing gene within the individual's body. Is it clear? So let me write it here. If disease causing gene is present within the individual's body, base pairing in between the single stranded DNA or RNA with the concerned DNA sequence, does not take place. And here appearance of base pairing and appearance of radioactivity nature, it indicates that there is no disease causing gene within the individual's body. Is it clear? So let me arrange the things here. So here, as already mentioned, appearance of radioactivity nature and base pairing in between the two consent sequences, indicates that there is no disease causing gene. Within the individual's body. And the other alternative strategy through which we can detect the presence of the disease causing pathogen at the early stages, here it is nothing but antigen antibody interaction. Is it clear? Antigen antibody interaction is the other alternative strategy through which we can detect the presence of disease causing pathogen. So here how the antigen antibody interaction takes place. So here most commonly antigen antibody interaction it takes place by taking the advantage of a specific strategy or a specific process termed as enzyme linked immunosorbent assay, which is simply abbreviated as ELISA. Is it clear? Antigen antibody interaction, takes place by the means of ELISA. ELISA stands for enzyme linked immunosorbent assay. So here how the specific ELISA it takes place or it occurs. So here initially let us assume it as the ELISA kit. So here within the ELISA kit, consent antibody it is successfully placed here. Is it clear? Within the ELISA kit, here the consent antibody it is successfully placed here. Thereafter, we need to insert the blood serum. So here within the blood serum, if there is an availability of disease causing gene, so here the specific disease causing gene or the specific disease causing antigen, it may remain in association with the consent antibody. Is it clear? So here after the successful formation of antigen antibody interaction, now here to the primary antibody, one more other secondary antibody it is successfully placed here. Secondary antibody, right? So here the consent secondary antibody it has a chromogenic enzyme. So to the chromogenic enzyme, when the chromogenic substrate is placed, here. So here in the presence of chromogenic substrate, the consent chromogenic enzyme, it is capable to induce color. So here appearance of color, it indicates that disease causing gene, it is present within the blood serum, and absence of color, it indicates that there is no disease causing gene within the individual's body. Is it clear? So let me write it here. In ELISA, initially antibody is placed within the ELISA kit. Right? So here to the consent antibody, a specific antigen may remain in association. And to the primary antibody, secondary antibody is subjected. Secondary antibody which has chromogenic enzyme is subjected. This chromogenic enzyme by taking the advantage of chromogenic substrate, chromogenic substrate, chromogenic substrate, it induces color. So here appearance of color, so let me write it here. Appearance of color indicates that disease causing gene is present. Disease causing gene is present. And here disappearance of, so let me write it here, absence of color indicates that disease causing gene is absent. Here, this is regarding role of biotechnology in medicine in detail. And in the next upcoming lecture session, we will be studying regarding transgenic animals. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.